Testing, 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 testing. 1, 2, 3. Can y'all in the back hear me? Not really. Can we go up somewhere? Testing, testing. Hey, Jackie, am I obnoxious? Perfect, right there. give a little recap real quick of what Tap Talks is. So you'll notice that we're going to start. Are we good? We're not yelling at anyone? Everyone looks, for those of you online, everyone looks terrified right now that I'm going to make the feedback happen again. All right, I think we're good. So I just wanted to give a heads up of what this is. So you will notice that we have trivia going on. It will be shown again later too, so there's no hurry to go through it. But we will show it between every talk that we have. There are prizes for the people who are here at Rahr Brewery. So whenever you're done filling it out, if you'll just turn it in at the back, you can see the guy waving at us. This is Logan. If you'll just turn it into him, we will score it at the end, and then we will find all the winners, and we do have prizes available. On the back, if you'll put whatever information you're comfortable. If you do give us your email address, we will email you about future events, but you don't have to. You'll still be eligible as long as you give us your name. We're going to start a few minutes late because this is the first time we have been here at Rahr Brewery, and the parking situation's a little confusing. There's plenty of parking, but finding the front door, I think we all had that experience. So we're going to start probably in about 5-10 minutes, but please feel free to do trivia. Another thing you're going to notice is that we have some people walking around. Ben, can you show your pin and your bracelets? So if you see anyone that is carrying these bracelets and this pin, if you go and talk to them about science or what life like a scientist is, all of these people are scientists, and whether they're industry or grad student or professor. And so you can talk to them about like what is it they do. They'll give you a bracelet. Once you get 2 of these bracelets, you can turn them in again at the back where Logan was, and he will give you a free choice of a sticker or a keychain. And we have a bunch of different science stickers that you can have. So yeah, that's basically what Tap Talks is. Like I said, we're going to start a little bit late because we still have people coming in. If you have any questions, feel free to ask anyone in one of these purple shirts, and we'll be happy to help you. So thank you all for coming today, and we'll get started in a little bit. Thanks. And once it's on, you are live. All right. We have a much larger crowd now. I'm very thankful. So we are going to go ahead and get started. Like I said, trivia will go again during intermission. So if you missed any questions, don't worry about it. It will happen. So I would like to introduce Dr. Cynthia Kilpatrick, or Cindy. She is a linguistics at UT Arlington, I believe, and has done some field work, which I hope you'll talk about as well. But if we could all welcome Cindy up to the stage, please. Perfect.
I'm going to stand over here. Can everybody hear me okay? Okay, so I want to ask a question to start with. You see my title. And I want to ask, when you see that first line, who thinks that that means we know a whole lot about language? Who thinks that that means we don't know much about language? So we are all correct. Because depending on how you say this, it might mean either one. I can say, wow, we know shit about language. And then it means we know a lot, right? Or I can say, we know shit about language. And then we don't know anything. So tonight, I want to convince you of both of those facts. We know shit about language, but we still know shit about language. Okay? So I'm a linguist. And anytime I go anywhere and I meet new people and I say I'm a linguist, guess what they ask me? What would you ask somebody if they said, I'm a linguist? What languages do you speak? Or how many languages do you speak? I'm a bad linguist, okay? I speak English sometimes. And I try really hard to speak Spanish. But as a linguist, I don't study languages in order to speak them. I learn languages in order to learn more about language. I study the science of language. So I want to know how language works, and I want to know how language is processed, what is going on in our brains when we are speaking. I want to know how language functions as a system, and we have all different cognitive processes. I want to know how language is different from other cognitive systems. I want to know how individual pieces of language work. And I want to talk about theories of language. I promise not to talk about theories tonight, okay? But I am going to talk about some experimental work that we do in order to figure out what people know about language. So we're going to talk about how we know things that we didn't explicitly learn. And we're going to talk about the fact that what we know is not always reflected in what we say, right? And then we're going to talk about how what we already know affects how we learn, and how what I know might be different from what you know, and that's okay, all right? So the first thing, when we think about knowing things that we didn't explicitly learn, we're really thinking about kids and how kids learn language. Does anybody in here except me have kids? A couple people have kids. Okay, at least there's somebody and I'm not alone. Okay, so take my word for it then. By the time kids go to school, they already know language. Have you ever spoken with a 4-year-old? Did they talk your ear off? Right? It wasn't like they could only say one or two things. They could say a lot because they already know how to express themselves with language, and they already know how to understand you. And as parents, we probably didn't teach them anything. I don't tell my kid, okay, now, when he's 2, we're going to talk about past tense because you have to do this a certain way. We don't do that. We just use past tense, and our kids learn it, right? Sometimes we teach our kids new words, but not explicitly. We don't teach them sounds. They just know these things. And if anybody has kids, or if you were a kid at some point in your life, you know that even if you try to teach kids things, they won't necessarily learn it, right? So here's my example. This is my husband and my son when my son was about 3 years old. And I had the distinct pleasure of listening to them have this conversation. So my son said, them are blocks and them are, and Dad interrupts. And he says, they are. And my kid thinks he's right. Them are. And my husband says, say they are. And my son dutifully repeats, they are blocks, blocks. Say they are blocks. And my kid says, they are blocks and them are clocks. Right? So sometimes we try to teach our kids something and they don't listen. So what we try to explicitly teach them just goes right over their head. So we know lots of things about language that we don't explicitly learn. So how do we know what kids learn? How do we know what anybody knows about language? So sometimes it's easy to think of biology as a science, or geology as a science, or botany or zoology as a science. But when we think about language, we think, how can that be a science? And here tonight I want to show you what we do as science in language. So we make hypotheses, we observe and analyze data, we run experiments, and one of the main things that we do is we collect language samples. So a language sample is, sorry, I didn't mean to do that. A language sample is where we bring somebody into our lab. It really is a lab. We have a room on UTA campus that is our speech sounds lab. And usually we pay them money because people don't want to come in a lab otherwise, right? And we ask them to answer questions or respond to things. And we gather data from them so we can learn more. And we do this with all types of ages. And what we've found is that sometimes what we know about language is not reflected in what we say. So here's an, am I hard to hear? Okay, so sometimes we make speech errors, right? So if somebody said to you, oh, I just saw a flutterby, would you know what they meant? Or would you think, oh, poor thing, you don't know what that winged creature is called? Right? You know what they mean. They just made a speech error. We assume they know that word, right? But when we start to dig deeper into looking at what people know, we realize that there are lots of ways that what we know about language is not reflected in what people say. So I've been doing this ongoing project with another professor at UTA where we have gone through, like, I don't know how many iterations of this. So first, we brought kids into a fully equipped play space. This was like the mecca of playrooms, okay? It had all kinds of toys. And for me as a researcher, it was awesome because it had cameras all over and it had microphones all over. And we brought the kids in with their parents and we said, just play with your kid. They can play with anything in this room. And our one request is that you just let us record you and videotape you. So the parents and the kids came in and they talked for 30, 45 minutes. And the only thing we asked of them other than to talk was, we said, at some point, can you talk about something that happened in the past? Some point, talk about something that happened in the past. They said, okay. So they leave, and we ask them to come back a week later. And we do exactly the same thing. And then we compare their samples, and what we see is that the language that people use in one sample, it's the same people. They're talking to the same person. They're in exactly the same room, and the same toys are available to them. But the language that they use can be vastly different in terms of morphosyntactic structure. And we said, wow, we learned something because we kind of assumed it would be very similar. So we learned some shit about language, right? But when we did that, it raised all of these additional questions about language that made us go, wow, we know shit about language. We have to continue this. So we started looking at adult language, and we analyzed the language of the caregivers. Also quite different. And then we said, well, maybe it's that it's kids. Maybe talking to kids messes us up. Parents, do you feel like talking to your kids has messed up your language? Sometimes I feel that way, right? So we said, well, maybe we need adults talking to adults. So we had adults do the same thing. We did not put them in a playroom, okay? But we gave them we gave them money for one thing, and instructions. And instructions to just talk to a friend for about a half an hour and record it and then send us the recording. And then a week later to do the same thing, same person, same place, same time, right? And it was fascinating to us because some things when we compared these samples were very consistent. So we have this thing called a multi-verb utterance. All that means is it's a statement with more than one verb in it, and everybody does this a lot. A 3-year-old does this a lot, a parent talking to a kid does this a lot, and adults talking to each other. But what was very different was the past tense and the plural. So even though we told parents to talk to their kids about something that happened in the past, in the language samples where adults were talking to kids, they almost never used the past tense. But in language samples where adults are talking to adults, they use the past tense a lot. So what does this mean? Maybe it means that as adults, we're interested in what has gone before. Think about what you do. You go see your friend, you say, hey, how are you? I haven't seen you in so long. What have you been up to? What did you do yesterday? What did you do last week? But this is not what we do with kids, right? With kids, we focus on the here and now. So we thought, well, maybe that's predictable, right? Other people have said that with kids, we focus more on the here and now. But what was unpredictable was plural. When adults talk to adults, we use plural a lot more than when adults talk to kids. I have no idea why. But if you have ideas why, please come talk to me, and maybe you can be our co-author, okay? Because what we realized is we know some shit about language, but when it comes to plural, we know shit about language. We don't know why they're doing this. We still have many questions to be answered. But that's not the only question still to be answered in language. One of my focus areas is second language acquisition. I look at how we learn second languages and what we do when we learn a language. And I'm sorry, but Pat, could you bring me my cup of water? Thanks. So, thank you so much. I am a language learner of Spanish. And as a language learner of Spanish, do we have any other Spanish speakers in the room? Okay, so I'm a second language learner of Spanish, and that means cuando yo hablo español, I sound like a white girl, right? Okay? I sound like an American. I have a very English accented Spanish. And there are certain aspects of English that creep into the Spanish of English speakers. And it's often related to vowels. So I speak with English accented Spanish. One of the things I do when I say a word like this, I say it like an English speaker. So I'm going to do a test on you. This word right here, the circled one, I don't want to say it and give you a hint. In English, how many syllables do you think this word has? 1, 2, 3, or 4? Hold up your fingers. Okay, I see 3s all over the room. That is exactly what I think, and that is what most native speakers of English think. Radio, right? 3 syllables. Spanish speakers. In Spanish, this exact same word, how many syllables does it have? 1, 2, 3, or 4? You guys are so good. Yes, in Spanish, it's radio. You got 2 syllables, that's it. Or, as an English speaker, radio, right? But it's not radio. And you might think, oh, well, that's because we have this same word in English, but that's not it. Because in English, we have that word, but when you give an English speaker a word in Spanish that doesn't exist in English, we do the same thing. So this word right here, to an English speaker, I will tell you from experience, English speakers say, tienda, right? And they make it 3 syllables. But Spanish speakers say, that's 2 syllables, tienda, right? So I looked at this in terms of all of these different sequences of vowels and how English speakers view those sequences of vowels in Spanish. Do we really learn anything about them? And for Spanish speakers, when they learn English, what do they learn about those? And what we realized was that maybe maybe we can take advantage of this for language learning. And so I worked with one of my grad students, Lori McLean Pierce, and we did this experiment on focused instruction in Spanish. And again, we brought people into the lab, and we paid them money, or we gave them credit in a class. And we said, okay, we're going to put you in a sound booth, and we're going to play a bunch of words in Spanish for you. And they would say, oh, we don't know Spanish. And we would say, that's okay. It doesn't matter if you know Spanish. We just want you to listen for syllables. And we had recorded a native Spanish speaker saying all of these words, so saying radio and saying tienda and saying these things in a purely Spanish accent that only has 2 syllables for those words. And all of these people would say they had 3 syllables, right? And so after they came out of the booth, we would say, well, we want to teach you something about Spanish syllables. So this word radio in English, it has 3 syllables, and we say radio. Well, how many syllables do you think that has in Spanish? And they would say, well, I thought it had 3, but since we're talking about it, it must not. And I would say, you're right, it does not. It has 2. So this has 2 syllables. How could you say this word with only 2 syllables? And they kind of play around with it, and they think about it. Radio. And they hurry up that last 2 syllables, mush them together. But that's what we do, right? We just mush 2 syllables together to make it 1 syllable. So what we found is that by helping people notice something that was different with their first language and their second language, it could help them learn their second language better. Because when we sent them back into the booth and we had them listen to those words again, they improved drastically on hearing how many syllables were in a word. So we did a scientific experiment to see if we could help people learn a language better. So we learned something about language. We learned some shit about language. But guess what? We still don't know shit about language, right? So another area where my language might differ from your language or different people's language might differ is when we speak a different dialect of English. So I am a proud Southern girl. I speak Southern English. Who is with me? At least some of you. Who is proudly non-Southern? Who does not speak Southern English? Okay, good. We have both varieties. So you guys are my research subjects. All right. So in Southern English, it is well known that we allow what's called a double modal. So a modal, it's kind of like a helping verb. It's could, would, should, may, might, must, can, will, shall. So in standard varieties of English, you can only have one of these in a clause. But in Southern English, we put several of them together. So it's perfectly okay for me to say something like, you might should have another drink. Southern English speakers, who thinks that sounds pretty good? Northern or non-Southern English speaker who says, that is totally ungrammatical. Yeah. See, this is how linguists do research. We do what's called grammaticality judgments. And we ask people, how does this sound to you? Does it sound grammatical or does it sound ungrammatical? Well, so to Southern speakers, you might should have another drink sounds perfectly grammatical, but to speakers of non-Southern dialects, it sounds horrible. But as Southern speakers, we also know that we can't just combine modals willy-nilly. We can say, you might should have another drink, or you might could have another drink, or she might would have another drink, but we can't say, you should might have another drink. Southern speakers, are you with me? Can you say you should might have another drink? Anybody? Okay. Whew. It's always nice when my predictions are held up in front of a crowd. Okay, you can't say you can, may have another drink. So I was a grad student in California, and I was one of the few Southerners in my department. And when I first started looking at this, what I was finding was that there was all this research out there done by non-Southern speakers on double modals in Southern English. And they were correctly describing how we could use it, but that's all they were doing. And I said to my professor, I really want to analyze and examine this construction. And he said, I think everything has been said about double modals in English. it was like he was saying, we know all the shit there is to know about double modals. And I said, I think you're wrong. There is a special meaning to a double modal in Southern English, and I haven't seen anybody talk about it. And guys, you should have seen his eyes go, something new that people haven't done? I want to hear about this. So I worked on this for a couple years, and ultimately what I claimed was that in order to use a double modal, you have to have volition. Sorry. Whoever the sentence is about has to be able to make a choice. So for instance, I can say to my husband, hey babe, you should have another beer. I'm going to be here a while, right? But to my kid, if I want him to go clean his room, I am unlikely to say, hey, you should go clean your room, because you know what? My kid ain't got no choice, right? There is no volition. However, my kid loves to play Fortnite. Is that just a kid thing, or do you guys play Fortnite too? Okay, it's not just kids. Everybody loves Fortnite. So if I want his room clean and he wants to play Fortnite, then I can use that to negotiate. And if he says, hey, Mom, can I play Fortnite? Then I might say, hey, you should clean your room if you want to play Fortnite. Because I am suggesting to him that I think that's what he should do, but I realize he might disagree. He might not want to clean his room, but I'm going to offer him incentive in order to do it. So through this project, I learned a lot of shit about language, but I realized in terms of dialects and us knowing things that are different, there is still a ton of shit to be learned about language. So we know shit about language, but there is so much more to learn. Thank you.
All right, we do have a couple of questions online, but before we do that, are there any questions of people here? Ben, we actually have a question from Ben. Okay, cool. New system today, everyone.
I'm the guinea pig, because I'm like a stooge. They planted me to ask the first question. This? This one? This one?
Oh, that's fun now.
Okay. So, thank you. This is really fun.
Thanks.
I have a question which is both about jamming syllables together and about plurals, actually.
Okay.
So, I'm
Wait, are you a linguist?
I'm Texan, as you can
Okay.
tell from my accent, right? Like,
Yeah, of course.
So I moved to Texas quite recently, and I noticed that in Texas there is a plural you'll, right?
Mm-hmm.
Like Z in German, like or in other, and that doesn't exist anywhere else in America. So why do we have that here?
Well, there's a gap in the paradigm, right? So you is singular and you is plural, and in order to tell them apart, we have y'all. Right? So otherwise, how do you know if I'm talking to you or if I'm talking to your whole group?
But my question is that, you know, the English language doesn't have a plural you.
Right.
But the Texan language does, and so does every other European language. And so is there a reason why we have it and the rest of English doesn't?
Well, despite what many people may claim, Southerners are super smart. So we have developed something that not everyone has. But I think... Sorry.
Maybe you should repeat my question.
Okay, so if you can't hear his question, he's asking why in Southern English we have y'all as a plural. Sorry. Y'all, when other dialects of English don't have a plural you. And there's not necessarily a good answer. I'm not a historical linguist. But certainly that has developed as a contraction of you all. And we use it like that. And I think Southern English, you know, think about double modals. We use this all the time, but when we learn standard English, they try to smack that out of us, right? My grammar teachers in middle school told me, you can't do that. But I do it, and it's meaningful. And maybe some other dialects more closely follow standard English than Southern English does. Maybe we just need it more, right? We're often in groups. We're talking in groups, right? That's a great question, though. I'd love to talk to you more about that in a little bit. Yeah. They might not. That was me. I was going to come closer so I could hear you.
It's really, it works, but it's super low.
Yeah.
Like this?
Yeah.
Test. It's
Okay,
not
so
making
she
any sound.
asked a great. Am I on
Test.
again? Okay, are we ready? Can you guys hear me? No?
All right, we're going to take a 5-minute intermission while we figure out our sound, everyone. Just 5 minutes. We do have questions, and I'm terribly sorry about this. 5 minutes. This is me. I am still on. Okay, so I am at least making some noise. Are we making noise? Can you make sure yours is still plugged? I'm sure it is, but just in case. We have not used before. And so things are a little bit different. I appreciate y'all's patience. Don't y'all remember how fun in-person things are? We finally figured out Zoom, and now we're not having to do Zoom anymore at least. So this is now where we're at. All right, speaker up next. Can we do hers? Hers is the most important. Yeah, sure. I think we got hers turned off somehow. So can you talk, please?
Okay, sure. Am I on? Can you hear me? Oh, I think I'm on again.
All right.
Okay. I'm going to just keep talking for a minute, and I'm going to address your question. Okay?
We just need the... We'll talk about that in a minute.
The question was, with currently what's happening with gender, what's going on with language and pronouns. And so what we're seeing is this addition of they or them as a singular pronoun versus it always having been a plural pronoun in the past. Now, curiously, we still use verb forms that go with the plural, even though we use it singularly, right? So if you are calling someone they, you still say they are my friend instead of they is my friend, right? But I haven't seen any research on that. I've just observed that. But there's research waiting to be done. We need to know more shit about language. Come study.
Check. 1, 2, 1, 2. Check.
Yeah. Oh, that's a great point. So she added a great point that we have gendered words that don't have an in-between like they. So you can have a niece or a nephew, but a niece is a girl and a nephew's a boy. And what do you call somebody who is non-binary, right? So I think that's going to change language drastically over the next few years. We're going to see much more gender-neutral terms coming in and being used in the language.
All right, I'm going to ask a quick question that was online. There were several questions of saying, wow, this is amazing. I want to be involved in these studies. How does someone even start getting involved in linguistics?
So anybody so as it happens, I'm the graduate advisor for our department.
Oh, perfect.
So
okay.
if somebody wants to talk about a graduate degree in linguistics, I'm the person they should talk to, and my email is right here.
This is perfect. For those online, in case it's not showing up enough, it is cynkil@uta.edu. Graduate assistant advisor?
I'm the graduate advisor.
Graduate advisor, sorry, for the linguistics department. So I saw several people saying that they were interested in it. So whether you're online or here, that'll be perfect. Do we have any more questions in the audience real quick? So as I had said, we have one over here, Ben. During the intermissions, you can go talk to people. Any of our scientists, you'll be here. So you can go talk to Cindy a little bit more after this and still get more information if you'd rather talk to her individually. All right, Miranda?
So, hello. My question was, when you were talking about the kids and the adult study, my first question is that true across multiple languages? Like, is it just that it's English that they don't use as much past or plural, or is it also across other languages as well?
That is exactly the right question to ask, but it's not a question we have answered. So far, we have only looked at English-speaking children and adults, but it would be great to be able to extend that to other languages.
Gotcha. Yeah. And then my other question was, do y'all know like why maybe they use past tense less? Is it because of the age difference?
So the kids at the age that we were testing, they should have already had command of past tense. They should have already learned it. But there's other research that says that anytime you have parents talking to kids, they're really focusing on what's going on in front of them right then. So they're saying, you know, this is a block, and this is a clock, or them are blocks and them are clocks, right? And they're looking at the things that are right there in front of them, and they're not thinking about these abstract concepts, especially ones that transcend time and space.
Gotcha. Okay, yeah. Thank you.
Sure.
All right, do we have any more before we do a short intermission? Do we have one more? Oh, what is it?
How long has they, them been used as a singular pronoun?
All right, so online, there's a question of how long has they, them been used as a singular pronoun?
Oh, wow. I do not know the answer to that question. It's been certainly much more prominent in the last year or two, maybe, but I'm not sure when it started. That is not something I know. I'm so sorry.
Not a problem. All right. So we are going to go into intermission. Those who are here or online, please feel free to get another drink. We will be back in about 15 minutes to learn about carnivorous plants. All right? Thank you all. Mup. By the way, there will be one more time of trivia, so please don't panic. We'll do one more intermission. All these plants are going to be part of the next talk. You're welcome to come look at them before the talk. I promise we'll do trivia again after this.
Okay, how's this sounding? Everybody able to hear this all right? Over in the back? Okay, all right, well, we got this.
Pocket or hold it or
Okay.
whatever.
Mic is on?
Do what?
Yep, mic is
Yep.
on.
We're going to talk a couple of minutes so you can adjust it. We're just talking
Perfect.
to each other.
Okay,
Yeah.
thank you.
All right, so we are about to start. Hopefully we have the audio a little better. If you have any issues at any point, if you'll just wave at me, we will adjust them, okay?
Yeah.
Am I too loud? Well, that's too bad because I'm not the one giving the talk. All right, do you mind talking, please, Paul?
Okay.
All right, can we hear Paul? He's the important one. Can we go up a little more, Logan? Keep talking.
Okay, I'm going. How's that, better?
Perfect.
Trust me, you get me started talking and I won't be able to shut up.
All right, so this talk is a little unconventional for Tap Talks. We are going to present up here. I'm hoping that you're able to see. You can probably lower me a little bit.
Nice there,
If you are wanting to see the plants a little bit closer, please feel free to come up. But if you are going to be too close to someone, please put a mask on, even if you've been vaccinated. Right now with the Delta variant, CDC does prefer to people have masks if you're without outside of your bubble. So please put a mask on and come up close. Otherwise, during intermission, we are going to move all these plants back to the back, and you can come talk to Paul again about all his plants, okay? But do feel free to come close. Just put your mask on. All right.
All right.
So I do want to introduce Paul, and if you will start talking about all your stuff.
Hello, everybody. My name is Paul. I run the Texas Triffid Ranch, Dallas's pretty much only carnivorous plant gallery. And today we're going to sit back and focus on a basic point on carnivorous plants. The first thing to consider with all of these is that there's an official definition of what constitutes a carnivorous plant. Not just the fact, okay, it looks gnarly, it looks dangerous. It really comes down to, do these plants have the ability to attract, capture, and digest insect and other animal prey? Now, the reason why that's important is that you have a lot of plants that see, right now there are between 800 and 1,200 species of known carnivorous plant. And it's determined based on, well, honestly, who's arguing with whom on any particular day. Because you have a whole group of plants that are also called proto-carnivorous plants. These are ones that have the ability to attract and capture prey, but they can't actually digest it. In fact, I'm willing to bet you. In fact, here's a quick call of hands here. How many of you know for a fact that you've eaten a carnivorous or a proto-carnivorous plant within the last week? I was going to say, if anyone was holding up their hand, I'd just say, quit pulling my leg. Okay, now here's the other question. The reality of it is that probably about half of you have eaten not one, but 2 proto-carnivorous plants within the last week. Hands up now, who has had fries with ketchup? Okay. Tomatoes and potatoes are proto-carnivorous. They have sticky hairs on the backs of their flower scapes and on their leaves that go ahead and capture insects that are trying to go ahead and feed on them. However, they don't have the ability to digest them. And so, therefore, they are considered proto-carnivorous plants. True carnivores are going ahead and catching their prey not out of malevolence, not out of any Little Shop of Horrors silliness, but solely because of the fact they live in environments that are very, very low in nitrogen, very low in phosphorus. And plants that live in very low nitrogen environments have one of 2 strategies. The first one is what mesquite, beans, and alfalfa do. They have bacteria in their roots that can convert atmospheric nitrogen into a form that the plant can use. It takes a lot of effort. They don't do well with a lot of competition, and that's why they thrive best in the low nitrogen environments. Then you've got the carnivores that let their nitrogen come to them in the form of insects, other prey. There are some exceptions in here. I'll go ahead and get to those in a minute. Now, obviously, the first and biggest one here that everybody goes on about is the old Venus Flytrap. Now, it's a matter of you find it all over the place. I can't tell you how many times people tell me, oh, I had a Venus Flytrap, but it died. They don't know why it died. And a lot of what it comes down to is that these guys really are some of the toughest plants I have here. Flytraps are great, don't get me wrong. But the problem is that they need rainwater, distilled water, because they're used to living in a really low nitrogen, really low salt environment. And the water throughout North Texas is pretty much crunchy. That's the best way to describe it. The first flytrap I ever had, I bought when I was in Chicago in 1979. I was 13 years old. It was doing great in Chicago. I moved to Flower Mound with my family at the end of '79. The first time I watered my plant, it was dead within an hour. And it took me 23 years to find out what killed it. And it was because the water in a flower mound is so crunchy, you can always tell who has sprinklers around their houses, because the side of the house and the sidewalk and the car and the cat and the kids are all stained bright rust red. So you've got that issue. Anyway, the flytraps here are just one of, again, at least 800 species known worldwide. There's at least 1 to 5 new species discovered every year. There's 1 new genus that's discovered about every 5 years. In fact, some of you may have seen the bits on the news about the new carnivorous plant that was discovered out in the Pacific Northwest. That is not only a brand new genus, it is a matter of this was a plant that was hiding in plain sight for decades that nobody had any idea was actually going ahead and capturing insect prey. Because you have some that will go ahead and are hiding in plain sight. You have others that are only carnivorous when they're blooming. And I'll go ahead and I'll show an example of one of these here in a minute. Now, to go ahead and start off here, after we get through with the Venus Flytrap, it's a matter of that you have the most of the ones that people will go ahead and hold their hands up that they know, okay, well, I know what carnivorous plant is, pitcher plants. Now, the reality of it is that there are 4 different genera worldwide that are commonly referred to as pitcher plants. And I apologize, everyone, about my having to go into the Latin on a lot of these names, because a lot of these species do not have a regular common name. And even if it is, it's really kind of contradictory. Because people, as I had several people coming over here, is, okay, this is a pitcher plant. This is a pitcher plant. These 2 are pitcher plants, but they don't even remotely look alike. And it's because they're not related. They all produce an individual type of trap called a pitfall trap, but they are otherwise all of these are completely unrelated to each other. You have right here, let's see, in fact, just as a little extra here, most of you may assume carnivorous plants are only found in jungles, in weird places in Africa and whatnot. Africa is actually rather depleted in carnivorous plants. You're going to laugh about this, but some of the widest variety in carnivorous plants worldwide, the best place in the world to see more individual genera of carnivores than anywhere else in the world is outside of Tallahassee, Florida. Seriously, I mean, I tell people, these pitcher plants over here, there's a nice stand of them right outside of the Tallahassee airport. That you've got an entire road that goes around Tallahassee, Capitol Circle. And once you get out of the airport, you can either go left or you can go right. If you go straight, you go into Apalachicola National Forest. Now, there's a great stand of these straight ahead. You got to drive through about 1.5 miles of alligator-infested bog to get there, but, that's up to you. Anyway, the North American pitcher plants are native to the Gulf Coast of the United States. The vast majority of them are found in Florida Panhandle, Georgia, Alabama, and Mississippi. There's one species that's known all the way to the Big Thicket in East Texas. It's the only pitcher plant species known, confirmed in Texas. So you got to drive a ways to be able to go ahead and see one of these in the wild. And then there's this little guy, which most people go, they cannot believe that this little guy, this is Cydia purpurea, the purple pitcher plant. It has a common name because of the fact that in spring they produce this deep royal purple color. These guys grow all the way up the coast, up into Canada. In fact, these are the flower emblem, the Canadian equivalent of a state flower for the province of Newfoundland and Labrador. And just as a little aside, for those of you who have either are not Canadian like me or happen to aren't familiar with Canada, I like to tell people that the weather in Newfoundland is a gosh number. -40 means the same thing in Fahrenheit and Celsius, and that's about 4 to 5 months worth of the winter. Thank you very much. So when I get folks who ask, oh, can I leave these outside during the winter in Dallas? I go, look, if they can survive a Newfie winter, they can handle anything. So anyway, the actual structure on the pitchers are what really determine the differences between pitcher plants. The North American pitcher plants over here, and this white pitcher plant is a great example, is that they end up attracting insects. Both they secrete nectar on the underside of the lid, and the lip itself fluoresces very strongly under ultraviolet light at about 830 nanometers, about the same as a rock lamp. To go ahead and look at fluorescent minerals, this glows bright blue. Now, this species here, the white pitcher plant, Cydia leucophilia, also really glows under moonlight. Under a full moon, they just glow. And in fact, because of that, you end up finding that they catch a lot of nocturnal insects, things like click beetles, moths, whatnot, that'll come in there. You can actually come out and check these out at night, and you'll see the eyes of wolf spiders fluorescing, because the wolf spiders are taking advantage of any prey that's coming on in here. And what will happen is that insects coming in looking for the nectar will find more nectar in little glands right along here. And they'll go ahead and hang on for dear life, trying to get down there. You'll actually watch wasps hanging on with 1 foot, trying to get down just to get a little bit more. Now, as you can see, if they move in the wind, they have a tendency to dislodge those insects. And so from here to about here is nice, slick, and waxy. Any insect that tries to climb out can't climb out. If it tries to fly out, the shape of the pitcher actually draws it down further. Now from here to about all the way down is lined with very strong, very sharp hairs. They're so strong that if you were to split that open and run your finger up the other way, it's like running your finger up a bandsaw blade. Do not ask me how I know this, please. Anyway, what happened, in this case, the insect has one of two choices. It can either try to climb up and tear itself to pieces, or it climbs down. And when it runs out of down, the plant draws up fluid into the column and goes ahead, drowns that insect, and digests it. And digestive glands along the inside end up absorbing the nutrients to go ahead and pass on to the plant. Now with the Asian pitcher plant, that's much the same way, except they go ahead and they produce traps off of these little ribs off of the tips of their leaves. And these don't have hairs on them. Instead, the whole inside is covered with a special wax. They grow in little hexagons that gum up insects' foot pads if they fall in. But otherwise, it's the same way. They fluoresce along the lid. They secrete a lot of nectar. This species here has two little fangs coming off of the underside. They're what are called nectaries. What they're for, nobody has any idea. But it's a matter of that you know that this species is happy when it looks like it's drooling venom off of its fangs. That's exactly what it looks like. So anyway, the whole inside of the pitcher here is lined with that wax. And instead of just water being drawn up in here, you have a special fluid that's built up in here. There's a combination of digestive fluid and there's a polymer in it. It feels like snot. And it really acts as a wet blanket. It's enough to go ahead and wear out the insect. It drowns, it dies, and the plant is able to digest it. Now, the problem with these guys, with all of these, and by the way, for all of you who've heard, oh yeah, you can feed Venus Flytrap's hamburger, pay attention here. The problem with most of these insects is they can't handle big prey all by themselves. They don't have teeth, they don't have food processors, they don't have big sharp knives and chainsaws, so they can't turn big chunks of meat into small chunks of meat. So many of these live in symbiosis with various animals. The North American pitcher plants half of the time are full of green tree frogs that will just sit there and they'll peek out with this, "Hi, how's it going?" Look. And then when they feel, they feel threatened, they go ahead and hide in the pitcher. You have ants that live with the Nepenthes here that go ahead and actually dive into the pitchers and tear apart big prey. Because any prey that's too big for the plant to digest will rot within about 3 or 4 days and can actually go ahead and cause damage. And in fact, when you feed hamburger to a flytrap, usually you're giving both too big a chunk and you're giving something that's really fatty. And flytraps don't have digestive enzymes to break down fat. They can only break down protein. So odds are you drop a chunk of hamburger in there, it's going to rot, it's going to kill the trap, and if you're not lucky, the rot will actually spread to the crown of the plant and kill the whole plant. So don't feed them hamburger. Anyway. Now, let's see, then we get in, I'm going to go ahead and I'm going to save the Australian pitcher plant for last. But just remember, this one has another pitcher plant structure on it. Same situation, drawing in insects via nectar, via fluorescence, and they fall inside. Now, there are some of the Asian pitcher plants that are no longer carnivorous. They found interesting strategies to get around it. There's one species where the traps act as a nesting site for one of the smallest frogs in all of Asia. The frogs, a full-grown female can sit on my little fingernail. The males are about half that size, but they've got this monster call like the coqui frog of Puerto Rico. 120, 130 decibels, ranges through the jungle during the rainy season in Borneo, and that goes ahead and draws in these frogs. The females lay their eggs inside. The tadpoles, which are pretty much microscopic, spend their entire life cycle inside of the pitcher, and the plant gets all of its nitrogen off of frog waste, off of dead tadpoles over the entire ecosystem is growing inside. Same deal with these. When I was telling you about the green tree frogs that are living in here, they're pretty much pre-digesting the prey for the plant. Plant doesn't care if it's being used as a toilet, just as long as it's getting the nitrogen. And there are some species of Nepenthes here that have now completely adapted toward being able to digest dung. There are several species that have pitchers that look like little toilets, and they secrete a nectar on the other side that's really thick, almost like apple butter. And it draws in birds, it draws in rodents, it draws in tree shrews. They get caught, or they come in, they eat that, they then crap in the pitcher. The plant gets its nitrogen that way. And there's even one species that is no longer fluoresces and no longer produces digestive enzymes and no longer produces nectar because its traps act as a roosting site for one of the smallest bats in all of Asia. The bats actually roost inside and the plant gets all of its nitrogen off of guano. So, go ahead and move on. The next group that I'm going to go ahead and point out are the sticky traps. And you've got two main forms of that. You have either your flypaper trap, like this Mexican butterwort over here. This is one of the world's largest. They produce big flat traps that are covered with glue on the top. They also smell a little bit like rotten mushrooms, so they draw in fungus gnats. When you come on up here, you'll actually see there'll be fungus gnats caught on the underside. And that's how the plant's getting its nitrogen that way. Then you have the sundews over here. Now the sundews have individual sticky hairs, and there are quite a few groups that have independently developed sticky hairs like this. The sundews, though, their hairs move. They're officially called tentacles. And what happens here is they secrete a glue at the tip called mucilage. It's a sugar compound, so it both smells sweet and it fluoresces really strongly under ultraviolet light. Insects figure, oh boy, quick, easy drink. They fly in, they get caught in the glue. Now, obviously, you can't see it from here, but if I were to stick my finger on here, well, you could actually see how far the leaf pulls away. It actually throws off a nice string of adhesive. So most insects like mosquitoes, fungus gnats, fruit flies get caught in that, and they realize they're stuck. And as they're fighting, it actually causes the tentacles to move a little bit. And this was something that was only really confirmed this year. Those hairs are microelectric generators. They produce enough of a microcharge that it spreads through what are called calcium channels through the leaf that set off all the other ones. So that as a bug is fighting with one individual leaf, it causes enough of a charge to cause all the other ones to converge. And there are some species where the whole leaf will wrap around and will drool on that prey. The whole leaf will wrap around to increase the amount of surface area that's exposed to the bug. And then after about 3 or 4 days, after everything else is digested, the tentacles will move back out of the way, the leaf will unwrap, and all that'll be left are the indigestible portions, the shell, fat bodies, things like that. Now this one here, this is a threadleaf sundew from the Florida Panhandle. In the wild, I've seen these things grow this high. It's just, unfortunately, in North Texas, you got to be careful because we can get so dry. I end up having to grow them in vases like this because it's the only way for them to be able to be protected from the south wind. So they get to be fairly good size, but like I said, they don't move. Now, believe it or not, even though the flytraps don't look anything like sundews, they're actually members of the same family. Because in this case, flytraps are just sundews that don't have sticky hairs on them. Their tentacles are modified into 3 little hairs on the inside that when stimulated by an insect, either 2 hairs at the same time or any 2 hairs within 10 seconds, it sets off enough of an electrical charge to cause the trap to close most of the way. Now, if the prey's too big, it's just able to go ahead and pull out and go on its own way. So you can stick your finger in there, and unless you leave it there for a week, it's not going to digest your finger. If it's too small, it escapes. But if it's just right, if you were the size of a housefly, you got caught inside of a fly trap, the best thing you could do would be to go ahead and hold still. Because after about, maybe 8 hours or so, the trap will gradually open back up. And the plant needs to have that because of the fact that the traps are also the plant's leaves. That's why, as fun as it is to go ahead and set the traps off with your finger and watch them close, that's one of the worst things in the world you can do to them because they require marginally more energy to go ahead and reopen than what they would get if they were just photosynthesizing. So if one trap gets sawed off accidentally, that's fine. But if you're repeatedly setting them off, it's sort of like holding you face down in a bathtub for about an hour and then wondering, why do you feel like dancing, you know? Anybody who wants to test this, please feel free. I'm up for experimentation here. So anyway, the fly traps take advantage of that same microelectric charge to be able to go ahead and close the trap. And sundews, if they get overstimulated and then the prey disappears, because you will end up, say, with birds going in and pulling prey off, then again, same situation, is that they'll gradually unfurl and they'll be taken care of. Now we're also going to get into a few varieties here that most people have never heard of. Like, for instance, the Bladderworts over here. Okay, how many of you are familiar with the horror writer H.P. Lovecraft? How many of you knew today was H.P. Lovecraft's birthday? Okay, how many of you knew that he was a relation of mine? If you knew, I don't recognize anybody here, so I'm pretty certain you haven't listened to me bore you to death with it. Relation on my mom's side of the family. And this species here of bladderwort is what are referred to as cultivars. It's sort of like when you go into a garden center and you see all the different roses with different names. People will go ahead and breed those roses for different attributes, either big blooms or exceptional smell or the fact that they'll run in runners as opposed to growing upright. And if you get them described through the proper association, that name, that cultivar name is yours. You can name it whatever the hell you want. And in the case of carnivores, you're now seeing a lot of people who are going and breeding interesting cultivar varieties. There's a whole load of fly traps that have all sorts of weird names. Werewolf, Jaws, Xenomorph. I'm I'm serious. I mean, some of them were, but there's a whole load of bladderworts that are named after H.P. Lovecraft characters. There's an Asenath Waite, there's a Cthulhu, and then there's the Mrs. Marsh, a Lavinia Watley. The Asenath Waite is especially fun because, well, that's my great-grandmother's real name. I'm pretty certain Lovecraft actually Tuckerized her and put her into the story, The Thing on the Doorstep. So anyway, so this one, the Asenath Waite here is relatively rare, but it's a great example of how the bladderworts work. Most people have never seen a bladderwort. Most people wouldn't know what to look for with a bladderwort, but they are the most common group of carnivorous plants worldwide. There are at least 200 species of them known. There are a lot more that are turning up because most of the time you only recognize them when they're blooming. The rest of the time they just have little scrub there. They mix in with everything else. And you have three different groups of them. You have your aquatic varieties, which are free-floating in water. They go ahead and catch prey that's floating along like water fleas and whatnot. You have your terrestrial varieties, which grow in boggy mud like these. And then there's one entire group that is only found growing on the rocks on waterfalls. And they all work the same way. In this case, I can't show you any of these because unless we had a microscope out here to be able to pull these off. But they produce these runners that have these tiny bladders at the end. Picture a clear tennis ball. It's been squished flat. It's got a slit in the side. It's been squished flat, and it's got some sensory hairs up on the top. The sensory hairs get set off. It causes the tennis ball to open up and slurp up everything in the vicinity. And then a valve closes, and it gradually spits all that water out and digests whatever's in there. And so these guys are feeding purely off of microscopic organisms. I like to call them guilt-free carnivores because you don't have to worry about feeding them. You never have to feed them. And they're feeding off of nematodes and other microscopic organisms. There literally are millions of nematodes and every handful of dirt in there. So you're never going to run out of food. All they need is lots of light and you got to keep them moist. And otherwise, just general appreciation. They do beautifully. So, as far as other oddballs, when I was telling you about proto-carnivorous plants, this one here may not look like a whole lot here from a distance, but this is part of a really big discovery from about 15 years ago. This is a trigger plant from Australia. It's a genus that's only found in Australia today. They've got some cousins in Japan and some cousins at the tip of South America. But today they're only found in Australia. And in their case, they're carnivorous only when they're blooming. They produce tiny sticky hairs like the sundews, except they can't move. And insects that try to feed on the backs of the flowers get caught in the glue. Now with the trigger plant, what's interesting here, and when you come on up, you'll get a chance to see this. They have 4 petals on them, 2 big ones, 2 little ones, and then in between the large ones is a column. It looks a little bit like the hammer on a gun. And it acts the same way. When an insect lands on the flower, that column whips forward and hits them with pollen in one particular spot. And then gradually, over the space of about an hour, we'll go ahead and cock back and wait for the next one. After they've been set off about 5 or 6 times, that column locks forward permanently. That male flower is now a female flower. So a bug that's been to a male flower has been tagged in one spot, flies to a female flower, and the pollen is exactly the right place to be received. Now, the frail trigger plants over here get their name mostly because their blooms are that small. There are a lot of other trigger plant species that produce much larger blooms. Some of them are so big and so ready to hit that the Ministry of Tourism throughout southern Australia has warnings up. Well, don't let small children play with the trigger plants because they can actually draw blood. These guys, you don't have to worry about it. But these are a big deal because these move faster than any other non-seed pod related movement in the plant kingdom. You can actually watch them here, you can watch them here. I had a friend who was checking one of these out. She set it off. She saw here, she saw here. She didn't see the interstitial here. She freaked out so badly she dropped the pot. Oh, no, I was going to say, that was a near miss. Anyway, the trigger plants are just one of Australia's oddities. And the suspicion is that there were a lot, based on pollen analysis, that there were a lot of carnivores both in Australia and also in Antarctica before it froze over. And that's where we're going to go ahead and we're going to finish up with this little discussion here involving the Australian pitcher plant over here. Now, as I said, it's not related to the other pitcher plants. It's not related to the South American pitcher plants of Venezuela and Brazil. These have no close relations left anywhere on Earth. Okay, last question here. Okay, how many of you are Doctor Who fans? How many of you are familiar with the character of Missy? Missy, yes. Okay. Missy is as close as I'm ever going to get to having my grandmother back. Let's put it that way. And all my friends go, this explains so much. For those who aren't familiar, Missy is a villain. She is a complete smartass. She is proudly queen of evil. And yeah, that sums up my grandmother to a T, and I miss her horribly every single day. But it was one of those, if my grandmother showed at my front doorstep, threw me a TARDIS key and said, oh, don't do anything I wouldn't do, I wouldn't be wasting my time going back to go see the Battle of Agincourt. I wouldn't be wasting my time going back to Woodstock. I'd be going back to Antarctica 30 million years ago with a camera, because everything I'd be taking a picture of would be brand new to science, completely unknown. And the suspicion is that Antarctica had quite a few cousins to Cephalatus. Today, the closest relations that this plant has to any other plant left alive include apples and roses. Oh, okay, sorry about that. Trying to keep the feedback down to a dull roar. Anyway, as far as those relations, all that is that Cephalatus' ancestors and the ancestors of apples and roses separated 90 million years ago. They have been separated since the dinosaurs were still just really getting up and going. And so the odds are pretty good that there were other species in Antarctica, it's just, well, they all froze. So anyway, I figure now that I realize I'm running out of time here, so I'm going to go ahead and open up everything. Anybody have any specific questions? Come on up here and I'll answer them as best as I can. I also have posters over here if anybody would like them. And for those of you who would like to go ahead and see more carnivores, I have a gallery in Dallas off of Spring Valley and Central Expressway. We're having an open house tomorrow night from 4 o'clock until 9. Obviously, you're more than welcome to come on out and take a look at the weirdness and listen to me yammer on even more. This is like you're going to get me to shut up. Anyway, but otherwise, anybody have a question? Thank you. I hope I wasn't boring. I had you laughing. I know that I had you laughing, so. Okay. Who has a question?
I have a feeling you're going to have a lot more questions when they can come and point at the plants.
More than fair.
I do have one quick question. We have one over there. But, how do you choose which ones are getting the, mesh lids on top? Are you afraid they're going to come out and attack your home or something?
No, most of it actually has everything to do with the humidity. For instance, the North American pitcher plants and the fly traps, you pretty much have to keep outside in full sun. They need 6 to 8 hours of direct sun. That's why a fly trap, a lot of people when they tell me, oh, I had a fly trap and it died, they don't realize they kept it on a windowsill or they kept it indoors. Unless you're raising cannabis, you're not going to have an indoor light strong enough to be able to keep a fly trap alive. And I'm serious. Let me tell you, everybody in the carnivore trade is so thankful for Colorado every single day because we grow so many of these plants inside that we couldn't grow even 10 years ago, just because otherwise, we'd be messing with halogen lights and threatening to burn the place down, and the police going, that's a weird looking light coming out of your window there. But even then, the problem is that fly traps and the North American pitcher plants, along with a lot of what are called temperate plants, have to go dormant over the winter. And they, if they don't get that dormancy, they will wear out and die. From about Thanksgiving until St. Patrick's Day, they'll stop growing, they tend to die back. The big freeze we had in February usually doesn't affect them all that much because they're used to worse. I mean, for God's sake, the purple pitcher plant can handle a Newfie winter. Texas is nothing. But anyway, they need that time going dormant because they need to go ahead and store up energy in preparation for spring. And then they'll bloom in spring, then go ahead and produce traps, and everybody's happy. The ones that have the tops on them are more tropical varieties. And these need to have a higher humidity. The Asian pitcher plant here is one of the particularly bad ones. A lot of you may go ahead and see Asian pitcher plants for sale at flea markets and garden centers, usually in a hanging basket. Problem is that unless you're prepared to go ahead and really keep them humid, then you're going to have problems with them because half of their moisture requirements come from moisture they absorb through their leaves. If the average humidity stays below 50%, they stop producing pitchers because they simply cannot pull enough moisture through their roots. Want to know what a Nepenthes root system looks like? Go clean your shower drain one of these days. You see that clump? Yeah, that's exactly what it looks like. I mean, very seriously, it's like, your significant other, 4 cats, that's exactly what this looks like. And I wish I were kidding. Again, don't ask me how I know these things. Please don't. My sanity can't handle it. Anyway, but if the humidity averages out below 30%, they just simply die. They can't pull enough moisture up through their roots to be able to stay alive. And here in Dallas and Fort Worth, we can go in the morning in the summer from 95% humidity first thing in the morning down to 7% in the afternoon. So if you're hanging them up in a hanging basket, odds are pretty good they're going to dry out completely, and the poor plant will, at the very least, stop producing pitchers. So that's why I've got to go ahead and keep the tops on, because I'm trying to keep the humidity higher so that you'll get more of the proper growth on them.
Gotcha. I do feel like Texas has been at the like 90% the last few days.
Oh, tell me about it. It's been wonderful for the Sarracenia, let me tell you.
I do think we had a question with Juan in the back. Is that correct?
We
No, we canceled our question.
Oh, okay. Well,
Perfect.
cool. Okay.
Always the best.
We do have
All
a
right.
question.
I don't know if we had any online, but either way, you're welcome to ask him online, and I'm sure that he will be happy to go back through and answer in a couple of days. Everyone who is here, we're about to move all the plants back to this table here, and you're welcome to come and talk to him during the intermission. So you are going to have
Please.
15 minutes. Please get another drink. This will be your final chance to see the trivia, okay? So we'll put it up. You'll get to see it this time, I promise.
Yeah.
So if you're here, please go ahead and do the trivia. Turn in your forms at the back. We do have prizes. We also have a random winner. So even if you think that these questions are completely off the wall, I didn't make them. I'll introduce the person that made them later. We do have random winners with prizes. We do have one more thing I completely forgot to mention earlier, and someone reminded me. So we also do a raffle prize. This has two things. If you like what we do and you want to support us, please consider buying some tickets. The prize is a little terrarium that has a availability to monitor your humidity and some other things, meteorological, weather
A
stuff.
hygrometer?
Probably.
Okay, yeah.
Yeah.
Probably
So-
be a thermometer and a hygrometer in there. Okay, cool.
So it's very, very cool. But there's also a way to get free tickets, because I know a lot of us are college students and stuff kind of sucks. So if you are one of the people who doesn't want to give us money, but you want to give us your support, please consider following us. You can either subscribe to us on YouTube, follow us on Facebook, Instagram, or Twitter. And each one of these 4 is a free ticket. So you can get 4 free tickets. Otherwise, the tickets are going to be 1 for $1, or you can get 6 for $5.
Okay.
And it does, like I said, we put it entirely onto keeping this program going. We are entirely nonprofit. So do please consider donating if you have the money. Otherwise, please at least subscribe to us. It does help us. But until then, we have 15 minutes, and then we're going to learn about galaxy formations, I believe. So
yes.
stick around. All right, thank y'all.
Well, thank
and also,
you.
can we please thank Paul? That was amazing. That was so good. Thank you.
Thank you.
Hey everyone, can I ask a quick question? Is there anyone who still needs any of the trivia questions? Can you please raise your hand very high if you need any trivia questions? All right, we have one. This is going to be the last round of trivia questions. They're going to go a little bit faster. When you are done, please take them to the back. Once we get to question 10, we will be in the next speaker. So again, please, this will be the last because after this talk, we will go straight into the trivia answers, okay? So this is also your last chance to do the Raffle Prize, which again is a little terrarium that has the whole little meteorology part in it. comment in the screen. There you go. Is that good? A little more up. Well done. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. More numbers. Can you flip slightly away from the speaker? That helps. All right. Can you test again? Okay. All right. You need to tilt the camera. Possibly. Sorry, I have a speaker on tape. I will adjust the camera as soon as we get started. It's on mine. Plenty of an angle. I'll get it in just 1 second. All right. So I want to welcome someone from TCU. We haven't had any speakers from TCU actually, even though we aren't sure. Dr. Kat Berger, and she is going to talk about the galactic competition for resources. So please welcome Dr. Kat Berger. Sorry, this one. If you haven't turned in your trivia forms and you haven't participated in Raffle and you want to, I do need you to get them back there now. This is your last chance. But welcome back to Dr. Kat Berger.
Awesome. Hi, everyone. Thank you. In the universe, there's 100 billion galaxies out there. Those galaxies have stars, they have gas, they even have exotic stuff like dark matter. In this figure over here in the background, I'm showing some representative examples of what some galaxies look like. You can see that some of them have different shapes, sizes, colors, all different kinds of things can be different for different sorts of galaxies. And we're going to talk about some of those differences today and why we have some of the structure that we're seeing. But first, we have to talk about home team. We have to talk about the Milky Way. Our home, the Milky Way, contains about 100 billion stars in it, and of course one of those stars is our own sun. Surrounding the sun, as you know, is Earth, Venus, Jupiter, all the things that we love, our home base. Now, this image that I'm showing you here is an artist's rendition. I would love to show you an actual picture of the Milky Way from this kind of perspective, but that's impossible. Why? Because we've only traveled to the moon. You have to travel outside the galaxy to get this kind of view. So how in the world did artists reconstruct a figure that would come in like this? Well, essentially what they did was they talked to astronomers, and astronomers collected as much information as they could about the Milky Way. And we looked at other galaxies, and we essentially got an artist to sketch it out like a sketch artist. And this is what we think our galaxy looks like. We think that it has these beautiful spiral arms. We think that it has lots of stars over here. And with all those stars, if every one of those stars has one, a few, maybe a dozen planets, is there life out there? I don't know. I know that there's life in here, so there's definitely life in the universe. But with that kind of odds, probably. Now, today what we're going to be talking about is forming those things, forming those stars, forming those planets. How do we form them? Well, it turns out that we form them out of gas clouds. Galaxies have a tremendous amount of gas in them, and that gas will condense, it'll rotate around. That gas that's rotating around will eventually come up flying out like a pancake, like a flying saucer. The part of the inside that rotating disk will group together, bump into each other, collisions and so forth. They'll stick to each other. They'll start to form little stuff like rocks, asteroids, comets, planets. You group enough of that gas together. If you get enough of it together, you can pack it so much that it'll start to exert a lot of gravitational pressure. It'll push in on its gut, and then its gut will become like really hot, millions of degrees Kelvin. So hot that it can take little particles like hydrogen atoms, bang them together to create heavier stuff like helium. Nuclear fusion process that's occurring in the sun. That's a star. our star is crazy. It has particles that are fusing together, million-degree Kelvin gas in each core. But in order to create the sun, in order to create the Earth, and everything in our solar system, everything, all the stars that we see, we need gas. We need gas clouds. So a healthy galaxy that's able to form stars and planets has to have gas. So I'm going to show you two mugshots of two different galaxies here. And we have one galaxy, the one here on the left. We have these brilliant blue colors. We have spiral arms. We have some reds, some oranges. I mean, it's just pretty. I like looking at it. Then at the same time, on the right, we have this other weird galaxy. You know, it's not usually something that you might think of, but that's a galaxy as well. It also has stars in it, dark matter, things like that in it. Why in the world are those galaxies so different? Well, it turns out that those brilliant blue hues, that comes from regions in the galaxies that are forming stars. And so if we look at a region of our own galaxies, the Sundews, which is a region of space where new stars are being born, what happens in star clusters is that you take a giant molecular cloud, and I'm talking enough mass to make 1 million stars, and you take that cloud will start to fall in and fragment, and a whole bunch of weird hydrodynamical instabilities will happen, and you'll get a whole bunch of stars. You'll get thousands of stars out of this giant molecular cloud. The stars that you get will have a distribution that's kind of like a handful of the supermassive stars. You'll get a decent number of the medium mass stars, like the sun. The sun's kind of like a medium mass star. And then you'll get a whole slew of these low mass stars. Now, the high mass stars, those are drama queens. They are bright. They light up everywhere. They're kind of like, look at me, look at me, look at me. They have surfaces that are that are 10,000 degrees Kelvin. And because they're so hot on their surface, they shine brilliantly in the optical color blue. And they just shine so brightly that they just overwhelm everything else. All those low mass stars, they're faint, they're little, they're fiddly, they don't really shine that much. They don't really cause a lot of attention. And so whenever we see a galaxy that has a lot of blue, that means that there's a lot of high mass stars in it. These high mass stars are interesting because since they're shining so brightly, they need a lot of fuel to sustain all that light that they're shining, which means that the nuclear fusion occurring in their core has to be running rampant. And with processes going at that speed or that rate, I mean, they just tear through their fuel. They just go right through it. It's kind of like if you had an SUV versus a Prius. The massive stars or the SUVs, they have a big gas tank, but they're so not efficient. Like, they can't go very far at all versus the Prius, small gas tank, and then you've got some fun electrical stuff going on there. They can go quite far very efficiently, right? These are the SUVs. Drama queens, they die very, very rapidly. They die in only 1 million to 100 million years. And at this point you're thinking like, okay, that's not that quick. But remember, we're talking about on the order of galaxy lifetime. The Milky Way is like, what, 13 billion years old? 1 million years, like what, 1,000th of that? So it's pretty much like snapping the fingers to the galaxy. Like it's nothing. Okay, so with that, brilliant blue, red. In order to have this, you need active star formation. The galaxy has to have gas to be healthy. So at this point, you might be wondering, well, how's our galaxy health going? Turns out that artist rendition I showed you, there was a lot of blue in there. So, all right, we're forming a whole bunch of stars, we're forming planets, we're good. Well, of course, that's not the full story. It turns out that our galaxy is doing okay now, but when we add up how much gas it has, and quickly it's making stars, it's going through that gas really quickly. And we think that our galaxy is now in a transition phase where pretty soon it might start looking like that red galaxy. It might not be forming stars and planets very soon. Now, how can we solve that? Okay, well, luckily, the Milky Way lives in isolation. It interacts with its surroundings. And so if it can a trapped material in it, it can actually refill its gas tank. So what does it do? Very good. It drops us in. There's lingering stuff out there from after the Big Bang happened. Galaxies formed, stars formed, gas clouds formed. Some of those gas clouds never found a home into a galaxy. Some of them are still out there. This is one of those gas clouds that's still out there and it's getting gravitationally pulled to our galaxy. This gas cloud has about 2 million times the mass of our sun and gas. So it can make 2 million solar systems like the one we're in right now. Just from this gas cloud. And this gas cloud is headed towards our galaxy, so it's headed this way. Of course, for the Milky Way to capture this gas cloud, that's not an easy process either. We know there's going to be obstacles in the way. So, what does this gas cloud have to get through? It turns out it has to get through the Milky Way's defensive hot halo. Okay, so surrounding our galaxies are very messy. There's a whole bunch of hot gas that surrounds them. When I am talking about hot gas, I'm talking about 1,000,000 degrees Kelvin. Could you imagine being a gas cloud in the middle of 1,000,000 Kelvin degree gas? That's ridiculous. So that itself can cause the gas cloud to warm up and start to erode and evaporate away. So that's one thing that's causing this gas cloud to deteriorate. Remember, we want this gas cloud to reach the disk because that's where the stars are forming. The stars aren't forming outside of the disk, so if it doesn't reach the disk, we can't use to make stars and planets. Okay, so we have this defensive halo. Now, interestingly, with this defensive halo, there's a couple processes that happen. One, it just heats up and causes it to evaporate. But two, this gas cloud is moving through the halo, and so an apparent headwind is created. So how does that work? Okay, so let's ignore astrophysics for 2 seconds and let's think about here on Earth. Let's talk about an example. Let's say it's a marvelously sunny day outside, and you decide to get your bike out. I mean, heck yeah, right? Nice day, no wind, nothing like that. You get on your bike, you start pedaling. What happens? What happens is that your face has the audacity to start running into the air particles. Your face starts smacking into the air particles, and those air particles hit back, right? And so now your face is getting hit by all this air, and air starts rushing past your face. You start hearing it howling against your ears and all those other things. Your hair starts flapping in the wind and all those other things. But at the same time, if you stop pedaling, there's no wind at all. And so when you're on a bike and you're pedaling, one thing that we should be very fortunate for is that our hair has strong molecular bonds. Because if it didn't, your hair would fly off, right? That would suck. We'd all be bald in 2 seconds. So fortunately, we have these strong molecular bonds. Now, we go back to our gas cloud. As our gas cloud is traveling through this hot halo, it's pedaling through the halo, and it's generating this apparent wind. Guess what? It doesn't have the strong molecular bonds that our hair does. The gas cloud just flies apart. And so it just flies apart because of the strong apparent wind situation as well. Now, let's zoom in so we can look at all the carnage. So here's this gas cloud in a close-up. I observed it with a green main telescope over here. This telescope is amazing. It's one of the best telescopes here on Earth. It's this 100-meter diameter. That's larger than the Statue of Liberty. It's awesome. So good. I had observed this gas cloud for over 100,000 observations to make this map. This is the most detailed map that we have for a gas cloud that is flowing to our galaxy to supply fresh material to it for future star formation. Remember that this is the leading part of the gas cloud where the gas cloud's going this direction towards our galaxy, and this end is the back end. And there's a whole bunch of chunks of it that are flying off. So this gas cloud's falling apart. Now there's a couple fun kind of weird hydrodynamical instabilities that I want to point out just because nerds can't help themselves. It's kind of like when you look up in the sky and you see these white sticky clouds, you start to see shapes and stuff. Well, we can start to understand what some of these shapes are. So, one of the cool shapes is that because that hot halo, very hot gas, very low density, very diffuse, not very much thick stuff there, but it's very hot. Now this gas cloud over here is only about 10,000 degrees Kelvin versus a million. So 10,000 sounds like a lot, but compared to a million, come on, that's very cold stuff. This over here is much higher density. What happens when you have high density stuff resting on top of low density stuff? Well, it's going to fall right through it. You know, it's going to ooze through it. So over here, we have these like ooze rocks that are essentially falling through the hot halo. And in addition, notice that these rocks are being pushed backwards in this direction over here. They're being pushed backwards because of that headwind. And these processes over here, these weird instabilities, are things that further cause the gas cloud to be disrupted. Okay, so we mentioned this one already. This is the drift and so forth. But all of this combined means that there's going to be less of this gas cloud to actually to start forming good. So how much? You know, if I was to kind of ballpark it, I'd say probably only about a third of it might actually reach the disk of our galaxy. And we study gas clouds like this all over the place. They're flown into our galaxy, out of our galaxy, all sorts of stuff to try to understand how galaxies evolve. There's a whole ecosystem in gas cycles. Now, let's kind of step back a little bit further. We've been talking about our own galaxy, but there's other galaxies out there. Let's look at our neighborhood. Okay, so near us, here we are right here. So you are here, and our nearest large galaxy neighbor is right there, Andromeda. Andromeda is not our closest galaxy, it's our closest large galaxy. This galaxy is about 2.5 times more massive than the Milky Way, but there's a whole bunch of little tiny satellites hanging around Andromeda and little tiny galaxies hanging around the Milky Way. A whole bunch of little ones. Satellites or dwarf galaxies, you call them both. Two of the larger dwarf galaxies that are hanging around the Milky Way are the Magellanic Clouds, the large and small Magellanic Clouds. Magellanic, Magellan. Magellan is the European person that discovered them. You can see these galaxies in the southern hemisphere. Why Magellanic Clouds instead of Magellanic Galaxies? Well, that's what they look like. Oh, it's probably not even hard to see it on this right here, but I think it still gets the point, is that they're just kind of misty little things. You know, they're things. To see them, you have to be in the southern hemisphere and go outside the city. You know, and so you'll see these little clouds up there, very cool stuff. And so what we're doing is we're observing these galaxies with the Hubble Space Telescope. We have a large Hubble Space Telescope grant that has just been awarded to us. I'll be leading this massive project, and people all over the country and the world. It's going to be very cool stuff. And we're also using other ground-based telescopes like this one over here. It's called the Wisconsin Stuff Mapper, WAM for short, kind of cool. And when we're looking at this particular galaxy, let's just zoom in on it, the larger of the two. It's got a bunch of stars in it, just like the other ones, just like the Milky Way does, just like any other galaxy. Now, let's take a look to see what's happening with this gas. Here's a picture of its gas, and this side over here is the observations, and this is a simulation of what we think is going on. Now, the observations over here, we have a whole bunch of these interesting circular splotchy things, and a whole bunch of these weird cells. What we think is going on is that, remember those massive drama-free stars we were talking about that don't live very long? Well, what do you think happens when they die? Just like any other dramas we make, whoa, they spew their guts out everywhere. They spew gas in every which direction. And when they're spewing their guts everywhere, where does it go? Left, right, up, down, side to side, everywhere, right? What happens when you get a whole bunch of these stars exploding? Well, you get gas flying everywhere, including outside of the galaxy, creating a galactic scale wind just from star guts everywhere. And that star guts contains a tremendous amount of gas. Those star guts, it's on the order of 10 million times the mass of our sun. So you can make 10 million solar systems. How cool is that? So what is going to happen to that gas? Well, guess who the neighbor of that gas or that galaxy is? The Milky Way, only one Milky Way diameter away. And our galaxy's evolving. You know it's going to trip that gas. It's going to pull it in, siphon it in, use it to make its own stars. Maybe it'll lose about a third of its gas. 5 fun, interesting, is that that's what the processes that's happening now. Our galaxy is such a ebullient that in about 4 billion years, it's not going to be satisfied with a simple siphoning, taking off the edges of it. It's going to cannibalize the whole. There's going to be a collision in about 4 billion years. We're going to combine. Our galaxy is going to get bigger and incorporate all of its stars, all of its gas, and take all of it. Kind of cool. All right, so let's go back to our mug shots of these two galaxies where we have the brilliant blue galaxy spiral, and then we have that red galaxy. Now, I've described to you what's going on with that brilliant blue versus that red, and we talked about, well, you have to have gas to get the blue. You know, you have to have star formation. That's needed. But the shape is also off. There's something very strange happening here. There's no spiral arms. There's nothing like that. There has to be more to the story. And so what that story is, galaxies have neighbors, not just gas clouds that cycle in and out, but galaxy neighbors. And those galaxies sometimes play gravitationally. They switch back and forth, tide, sometimes coalesce, sometimes merge. Here's a simulation of two galaxies that are roughly the same mass. Notice that when the intensity becomes close, all of a sudden you get a whole bunch of bright, brilliant blues occurring. Well, that's a big burst in star formation. A whole bunch of gas clouds are crashing into each other. Interestingly, the stars are so far apart in galaxies that when you have a galaxy collision, the stars completely miss each other, but the gas clouds collide, and that collision between the gas clouds causes them to compress, and it just triggers star formation out of the logic. So to get to that weird red shape for that galaxy, that weird kind of spherical thing, what happens is if you take some brilliant galaxies with blue ones, and you take them and you let them compete with each other gravitationally in a little fight, they might combine. And when they combine, they'll merge together, they'll trigger a bunch of star formation. And when they trigger a bunch of the star formation, well, guess what? You're going to also have those drama queen stars that don't live very long. They're going to die. They're going to start exploding. They're going to start kicking gas out everywhere. Now all the gas is going to get thrown out of the galaxy. And so now the next generation of stars might be a little bit more difficult to form unless you can catch that gas again. If you can throw a pencil in the air and catch it again, you know, you're good. But if you throw that pencil too hard, it can just fly out into space. So it depends on what this winter wind process. Sometimes you're okay, sometimes you're not. Sometimes a neighboring galaxy will be a bully and just grab it away from you if you're too close to them. And then also, you get processes like here where the gas is going out, like we were just describing. This is kind of like the aftermath of this. And then you also just get those tidal tails where material will just get pulled out and just flung out. Well, the thing is that once the gas is pulled out, it's not inside the disk anymore, so it can't really make stars. So it's less useful for the galaxy. And then through all these kind of processes, you think you get to something that looks like that. And so what I do is I study this whole entire gas cycle. I look at the gas that's inside the galaxy, that's around the galaxy, to try to understand what's going on with them. And I've just been talking about our closest neighbors. We have, that didn't work. We have a whole plethora of galaxies. Here we go. We have a whole plethora of galaxies, even, just outside our very, very tight local neighborhood. This is so close by stuff over here. And this is a simulation based off of actual observations. Because we can't fly through space. That'd be anything if we could. But based off of the actual observations, this is the distribution of where we think galaxies are. Notice that there's some bluer ones, there's some redder ones, there's some that are hanging out by themselves, there's some with buddies, there's some that have a lot of buddies. All of those other things shape them for what they are. And all that will affect whether or not the galaxy is healthy with gas on top of it and is able to make stars or not. And so that's what I do. I look at the health, the star formation health, and planet formation health of galaxies. And with that, I'd like to open it up for questions. Okay,
are they usually from, like, as you mentioned in your presentation, like star formations that ejected gases from galaxies or rogue sources typically from?
Yeah, okay, so when we look around galaxies, there's a lot of gas clouds. And so we do a census of trying to figure out exactly where these gas clouds came from. Some of the gas clouds that are out of galaxies are just like if you were to throw a pencil in the air, that pencil will fly up into the air, fall back down. Well, the same thing happens with galaxies that are very massive. The Milky Way, if you have a star that explodes, that explosion will kick gas out of it, but the gravitational pull of the Milky Way is so strong. That is a massive galaxy. That gas might get a little bit, hover above the disk for a little bit, but it's going back down. It's not going to escape. Now, for the Magellanic Clouds, because they're so much less massive, you don't have to throw as hard to get it to escape. So in some cases, it's from the galaxy itself. In other cases, galaxies rip apart neighbors. They'll just pull material off a neighboring galaxy, especially if you have a small galaxy that's near a large galaxy that's hanging out, that's traveling through that hot halo that we were talking about. That makes it very easy for tidal processes to affect any material that gets pushed out of that galaxy from any stars exploding. So it's kind of like a 1-2 punch. The small galaxy has a star, that star dies, it explodes, that material gets kicked out, and then the tides from the larger galaxy wash it away. The last scenario is that when the universe formed, it formed galaxies, it formed stars, it formed all those things, it formed particles. Some gas clouds that formed never found a home in a galaxy. They're just lingering. They're just the dredges, the leftovers, stuff between galaxies. Well, that stuff between galaxies is matter, and matter is attracted to other matter. So it'll slowly flow onto the galaxies. Now, of course, the leftovers don't last forever, so there will become a point where galaxies will start to become an even more crisis because they'll have a source of gas that will be lost. So as the universe ages, they won't be able to use that as a source forever. So galaxy cannibalization for the win.
Perfect. Are there any more questions? Oh, we have one right over here from Wilder.
I probably don't need the microphone because I'm pretty loud, but so when you have that diagram of the two galaxies merging and all that, really a simple question. I just was wondering, what is the typical time scale of that happening?
Oh yeah. Okay. So the time scale. All right. So the Magellanic Cloud is going to run into our galaxy in about 4 million years, and the same time Andromeda is going to run into our galaxy. Probably for the entire collisional process to finish, it depends on the size of the galaxy. When the Magellanic Cloud runs into the Milky Way, it'll probably occur pretty quickly because that's such a small galaxy. It has 1/100th the mass compared to the Milky Way. It'll get absorbed really quickly. So it'll probably be like, I don't know, 1 billion years or something, maybe 500 million years. I know, exactly, something like that. That is fast, I know. Right. Now, I wouldn't be surprised, though, if when Andromeda hits the Milky Way, it'll probably take a few billion years. So 2, 3, maybe, I don't know. Because it's tricky because it's like, when do you call it a settled galaxy? I mean, so it'll be like causing weird morphological stuff way before it interacts. But if you're just talking about the actual collision and mucking and merging and blah, probably a couple billion years, but quite a bit leading before that, and then there's going to be definitely some reverberation settling that'll happen for many billion years beyond that.
All right, I think we have another question right here in the back.
All right, testing 1, 2. Okay, so first off, awesome dress.
Thank you.
Second, what's your personal opinion on, if you had mentioned matter being attracted to matter, right?
Yes.
What's your opinion on dark energy acting as a negative pressure against gravity? Would the acceleration of the universe be an argument for, would that be an argument for the expansion of the universe or an expansion of dark energy?
Okay, so that's a tricky thing. So you mentioned in comparing it to gravity. So with gravity, matter and matter attract each other. So that's an attractive force, stuff coming together. But he was talking about a repulsive force, dark energy, which is this weird exotic stuff in the universe that astronomers and physicists are kind of not really sure what it is. We have some ideas of what it could be. There's a number of different theories. But what is occurring is that when we look at galaxies in every which direction, the ones that are kind of spaced out away from each other, they're slowly drifting apart from each other. That doesn't mean that if you were to look at the solar system isn't drifting apart. The Milky Way galaxy itself isn't drifting apart and spreading out bigger. It turns out that gravity is a much stronger force and it can keep things cohesive and together just fine. But that's only if they're next to each other, because we know gravity works as the more massive one object is, the more massive the other object is, stronger the pull. The closer they are, the stronger the pull. You take massive objects and you separate them out the wazoo, well, that gravitational force will become weaker. And there'll become a point where this very extraordinarily weak repulsive force will dominate. And that's what's causing the universe to expand. So it's not regions where galaxies are grouped together, where there's galaxy groups, clusters. They're not spreading apart. But a galaxy group and a galaxy cluster that's far away from each other, they're spreading apart from each other. And so that's what's happening with that. I don't know if that quite answers your question on that. I can talk more about what one of the ideas of dark energy is, but I don't know if that's too tangent.
All right. Do we have any other questions before we move on real quick? All right. Can we all please give Dr. Kat Berger a round of applause? Thank you.
Yay! Go Horned Frogs.
All right. So we are going to move right into raffle prizes and trivia. So first off is Raffle Prize. So if you did participate, we do greatly appreciate you. And I shook them up earlier, but I guess I could shake them up just in case y'all wonder. All right, we have 258-6805. If you can check your raffle ticket, please. It's 258-6805. I have the patience of non-existent, so I appreciate your support. Next is 258-6795. All right, we have right over here. Thank you so much. I appreciate it. You can go to the back and you can get the little terrarium. Thank you so much. All right, so for this next part, this is actually my last time that I will be helping lead TAP Talks. I am going to be handing it over to Ben Smithers. So I really do appreciate everybody who has supported and helped me lead it for the past 3 years. But I do hope to graduate at some point and move on. So I know it's been a while, but this is the final part. I would like to actually start writing my thesis. So for this next bit,
But first, can we get a round of applause for all the work Leslie's put in for TAP Talks over the years?
Greatly appreciate it.
Thank you, Leslie.
So I am going to have Ben go through all of the trivia. I do want to say he has been a hell of a supporter, and I truly believe he will get TAP Talks to continue going. He did support when he first joined. He helped me get the trivia going. And then whenever we went virtual, it literally would not have happened if he had not taken over and figured out how we could go virtual. And now this hybrid method, this whole time he's been going through, he's been making sure we could stream it. So I do want to say thank you, Ben, and he will be the one going through all of the trivia. And thank you all for all your support.
All right. I always love going over the trivia. I've written a lot of these, a lot of help from Nick over there. So I'm always really excited to get to go through the trivia because, you always run into fun things like doing the research, finding these weird bits of information. Some of them a bit weirder than others. But we're going to start at the top with trivia question 1, talking about Dr. Henrietta Leavitt's work towards establishing these things called standard candles to measure distances in astronomy. And so what we use to do that is these stars called Cepheid variables. That's the answer here. They kind of vary in brightness, and we can use that to derive a relationship there between how bright they are and figure out how far away they are. So moving on to question 2, what translated from French did CERN originally stand for? It is not named after Philip G. CERN. No, the answer is actually the European Council for Nuclear Research. So it's A. Now for question 3, how many babies does the official state animal of Texas have in a single litter. This one's a bit of a tricky one because you kind of need to know what the state animal of Texas is. It's the 9-banded armadillo, and the fun thing about it is they always have exactly 4 babies in a litter, and they're all identical. Yeah, I know it's shocking. That's how the truth always is. So we're going to move on to question 4. This forest-dwelling animal is one of the 2 members of the Giraffidae family, the other being, of course, the giraffe. And this one is, although I love the name of the common pronghorn, that's actually a North American creature, and we're talking about the okapi here. So, moving on, I really like this question too. I like them all, to be honest. So this material, seemingly straight out of Star Trek, because it actually is in Star Trek, is actually real. And you're already familiar with this if you've seen Star Trek IV, The Voyage Home. I'm talking about transparent aluminum, yeah. I don't know if we use it in spacecraft construction these days, but it is something that's real. We're here at Tap Talks to have our minds blown. So, question 6. Discoveries in science are sometimes misattributed to whomever popularized them rather than actually made the discovery, although named after Enrico Fermi, who actually invented Fermi's golden rule of quantum mechanics. Now, although I'm sure you all wish this was Bill Nye the Science Guy, which would really make a lot more sense as to like why we all talk about Bill Nye the Science Guy. The guy didn't actually do that much science as far as I know. This is actually Paul Dirac, but we don't hear about him on TV much, so that's a bit of a disappointment. For trivia question 7, the most distant galaxy in the known universe is observed as it was 13.4 billion years ago. Which of these groups of letters is it? Now, it's not the LA galaxy. That's only a couple thousand miles away. Let me double check to make sure I know the right letters here. This one actually is getting me a little confused. It is. Yeah, you're right. GNZ11. That's the Ursa Major. Now, this was also written about a year ago, maybe in, it's now 13.4 and 1 billion years ago. But so the whirling disease, which causes some fish to whirl forwards in an awkward corkscrew-like pattern, affects which kinds of fish? Is it flounder, eels, trout and salmon, or actually crustaceans? It's not actually crustaceans. We're going with trout and salmon. So we always like to have a snail question in Tap Talks too. It's been a while, honestly, since we've had one, and I feel sorry we've been slacking on the snails. So this is kind of actually terrifying, knowing that one of these is actually correct here. So do snails have beaks, a simple maw through which food passes, teeth, or probosces? Snails have teeth. Look up a picture of a snail's teeth. It's scary. There are just rows of them like a shark. And lastly, the 10th question here, the most abundant element in the Earth's crust. I know a lot of you might be thinking, all right, iron, nickel core, so it's probably iron or nickel, but a lot of the mantle and the crust is all made up of silicon dioxide. That's 2 oxygen. It's oxygen. We breathe it in the air, and we walk on it on the ground. And so, with that, we've made it through. We've exhausted the questions. And so, we're going to move on to a tiebreaker part. We had 3 people actually tie for first place with half the... Oh, this is the wrong group. So I'm going to need to call up 3 people to the stage. I'm going to list your names. And then we're going to do a little tiebreaker question where you all have to try to guess a little quantity as best as you can. So could I have... This is the hard part. I don't like paperwork. Phil, come up to the front. Ivana Moya and Miranda Williams. Come on up to the stage. Oh, I actually needed that paper that I just folded up and put away. Yeah, Phil. The thing is, I think we made the trivia a little hard this time. So, this is the way it works. I'm going to ask you a question. You're going to have to answer in a quantity. And the 3 of you will have to give a unique answer. And then whoever gets the closest gets to get the first pick of the prizes at the back there, over the table where you turn in the trivia forms. And then whoever got the second closest to the real quantity, you get to go in the back second, and you know, and so on and so forth. So the question today is, how many bananas would you have to eat to receive a potassium 40 dosage exceeding the radiation worker safety limits in the US? Yeah, just give it your best guess. Would you like to make a guess?
No.
Are you just going to concede?
Also no. 3,697.
3,697.
I was going to guess 450.
450 bananas?
I'll just do 100. I'll go low.
You're all making this pretty easy for me. It's all in descending order here. So you would actually need to eat 500,000 bananas to get enough potassium in your body to get, you know, a stern talking to from your boss. So, Phil, you get to get your first choice, and then Miranda second, and Ivana third. Can I get a round of applause for our first, second, and third winners of the trivia? Thank you for playing. Now, there's still two more people. And for fourth and fifth, you get a choice of Tap Talk's swag. So we do have these T-shirts here, very nice and stylish. We also have pint glasses with the same logo on them. And so for our fourth and fifth winners, we have Omar Yonori, and Maya Boyd. So Maya Boyd and Omar, you both have gotten fourth and fifth places. You both tied, actually. And so you can head on down to the back and get your pick of the Tap Talk swag. So, I know. Before we're done, we have one more winner for the trivia. And that is, every time we like to just grab a random name out of the list of people who submitted trivia forms. And so I was handed a form before I came up here, and I have someone's answer sheet, I guess, in my back pocket. And so I'm going to just draw that, and we're going to find out who the winner is here. Juan Cardenas. Congratulations. So that is the trivia for today. Thank you all for coming, and hope you had a great night. And next month we'll be over at Legal Draft in Arlington. So be sure to follow us on Facebook, Twitter, all the social medias, and we can't wait to see you again.